In this lecture, I'm going to be talking about a way of measuring functional connectivity with MRI. This is the first part, answering such questions as what is functional connectivity MRI and how do we estimate functional connectivity? There are different types of connectivity that we can measure with MRI. First, there's structural connectivity, that is how are different areas of the brain structurally connected, through the white matter fiber tracts. Second, we could look at functional connectivity. That is, how are different areas of the brain functionally related to one another? And finally, we can look at affective connectivity. That is, what is the influence of one or more brain regions on another? Essentially putting arrows on those functional connectivity. In this lecture, I'm going to be focusing on functional connectivity. That is, how are different parts of the brain functionally related to one another? In a typical task activation study, as for example here in this bilateral finger tapping task, we have the person perform some sort of task, and then we have our expected response, and we look for where, in what voxels do we see an expected response. Increases in the signal when the person is performing the task and decreases when they stop performing the task. For functional connectivity, Carl Friston earlier in fMRI defined functional connectivity as the temporal correlation between remote neurophysiological events. That is, if two parts of the brain, if we see the signal in two parts of the brain go up and down together, we call them functionally connected. Think of it as a functional co-activation. That's really what we mean by functional connectivity. So in this case, perhaps not too surprising, if we ask someone to tap the fingers on both the left and right hands at the same time, the left and right motor cortices are going to be functionally connected to one another. What is interesting is that we see the same thing at rest. So if we scan a person at rest, and if you look at the voxel in the motor cortex, this is the signal change that you might end up with. And if we ask what other areas in the brain have similar fluctuations, this is the map that we come up with. As you can see, looking very similar to that motor cortex map that I showed on the previous slide. And if you overlap these two fluctuations, you actually notice that there are some correlations, that there are some similarities between these fluctuations. So the idea is, in addition to task-related neural activity, there is spontaneous neural activity that co-activates within these different networks. These also produce bold responses that we can detect with MRI. This measure of resting state functional connectivity was first discovered by Bharat Biswal at the Medical College of Wisconsin in 1995. So in this study, he had individuals performing a bilateral finger tapping task. You can see the image of activation right here in that task. And actually before that, he acquired a scan at baseline, and then he looked for the correlations within a voxel, a seed region within the motor cortex. And you can see the striking similarity of these correlations obtained purely at rest compared to the actual activation map that was obtained during the finger tapping task. He actually performed a lot of controls of this as well. So initially, when he recruited these subjects, he said he was going to be doing a visual experiment. And he actually collected initially a baseline scan, said he was just adjusting the scanner at that point. And then he did do a visual experiment. And right at the end, he said, oh, by the way, could you also do some finger tapping task? And acquired the image that you see on the left. So in general, the person wasn't primed to try to think about tapping their fingers. He also measured with EMG electrodes, making sure that they actually weren't tapping their fingers. And yet you still see these correlative fluctuations at rest. The other thing that Bharat Biswal showed in this study is that these correlative fluctuations occur at very low frequencies, typically between 0.01 and 0.1 hertz. So these are cycles of about 10 to 100 seconds. And there's a lot of really great follow-up work that was done in the following years by Mark Lowe, Dietmar Cordes, actually right here at the University of Wisconsin-Madison. If you look at the early history of resting state functional connectivity MRI, as I mentioned, in 1995, Bharat Biswal published his paper showing the first demonstration of this resting state connectivity. A few years later, Mark Lowe here at the University of Wisconsin-Madison demonstrated that this effect could be seen outside of Milwaukee, and it could also be done on clinical equipment, a whole brain with a long TR. A year later, there was the first demonstration outside of Wisconsin. And then there was a lot of other great follow-up work showing that it's not just motor cortex, there's other networks that we can see with these resting state functional connectivity. And in fact, these fluctuations are even present on top of the task. What's interesting is that if you look at the first 10 publications of resting state functional connectivity, 9 of them come from Wisconsin. I put Mark Lowe's as in Indiana in parentheses because while he was in Indiana when the paper came out, this was actually research he performed while he was here at University of Wisconsin. So quite a nice history of Wisconsin in this field of resting state functional connectivity. And in fact, there are a number of resting state networks that can be reliably observed. This is an image from Christian Beckman showing 8 different networks. I've sometimes heard this referred to as the Beckman 8. That consistently are observed across individuals. And similarly, some studies from DeLuca and Demoiseau also showing similar reliable networks that can be observed across individuals. So how do we measure functional connectivity? I say in general, there would be 3 approaches: seed-based correlation, independent component analysis, and graph theory. Seed-based correlation is what I discussed previously. That is, we pick a particular seed region of interest, take the time course from that, and then we calculate the correlation of that time course with all other brain voxels. We get a correlation map, and that's what we call a connectivity map. A second way is to use independent component analysis. That is, if we sort of unravel our image and put all of the voxels along a row here, and the time courses along the columns here. So this is, if you think of this as a matrix of different time courses across all the different voxels in the brain, we can essentially decompose this matrix as certain components in time multiplying certain components in space. Or thinking of this visually, we take our data, that is a series of volumes over time, and we decompose them into different spatial components that share a particular characteristic time course. So all of these regions would share this particular time course, these regions would share this particular time course, and so on. And finally, we can look at this as a graph. That is, we can take a priori regions of interest, and this could be either a parcellation of the entire brain or just a subset of some of the regions in the brain. And then we calculate all possible pairwise correlations between these regions. And in this particular case, the areas that have significant connections or strong connections between them have lines connecting them, and the width of the line is representing how strong the connections are. And this is a study from Damien Fair where he was comparing the functional connectivity in children versus adults. And you can see that during development, a lot of these connections, in particular, the more anterior-posterior connections in the default mode network, are formed later on as you get more closer to adulthood. This idea of representing the network as a graph is actually really useful because there's a number of tools that have been developed to try to understand these graphs. It's what's become known as graph theory. And this has been applied not just to the brain, but also to other kinds of networks, the internet, social networks, road maps, and in our case, the brain. And there's different kinds of measures that you can calculate by representing the networks as a graph. Graph consisting of a series of nodes, that is regions, and edges, the connections between those regions. So you can look, for example, at functional segregation, that is how clustered are different regions, how modular is the brain. We can look at functional integration. So for example, what is the path length, which is how many hops does it take to get from one node in the brain or in the network to any other part of the node? And that's related to the efficiency of that network. We can look at the functional influence, that is how important is a particular node. We can look at how connected each of the nodes are, for example, degrees. And we can also look at how important these connections are. So there are a number of different kinds of metrics that we can calculate from these graphs that provide some additional useful information. What's kind of interesting is that one of these measures, the eigenvector centrality, is also related to the Google PageRank Index, which is how Google decides what web pages are most important and lists them to the top of the search criteria. So I find that interesting because in some of the critical ways of how the brain is organized is also how Google has found out is the best way to present relevant search results. So if we look at the number of publications per year using resting state functional connectivity, it started first in 1995. There's a few small handful of papers, and then really it just took off in the mid-2000s. And now you're seeing thousands of papers being published each year using this particular technique. And this has been applied to lots of different disorders, pretty much any mental disorder or condition people are looking at using resting state functional connectivity. Now, one of the questions you might have is, if it was discovered in 1995, why did it take over 10 years before this really took off? And I think the answer to that is in part, early in like the mid 90s, we were still grappling with questions such as, can we detect this response to very brief stimuli? Is the bold response linear? What are the properties of the bold response? How do we best detect it? And so, for someone to come along and say, well, we can also not do a task at all, we can just look at these spontaneous fluctuations, there was a lot of skepticism initially in the field. And so in these first 10 years, there was a lot of really good, solid foundational work being done to demonstrate that this resting state functional connectivity signal really is real, that it is really neuronal in origin and reflecting these spontaneous neuronal fluctuations. Perhaps another reason why we see this taking off is a set of studies that came out in the early to mid 2000s. One of these was a study from Mark Rakel entitled A Default Mode of Brain Function. This was actually a PET study, and Mark Rakel noticed that across a large number of different PET studies, that there are some regions that showed decreases in activity when a person engaged in some cognitively demanding task. At first he kept filing these away, and after enough of these, he realized, well, maybe there's something to this. If you look at the graph here, note that the reddish areas are actually showing the maximum decrease when a person is engaged in a particular task. And so we reasoned that perhaps these regions go down in activity because they're more active at rest. That is, they're essentially your default mode of your brain function while you're lying there at rest in the scanner. And a couple of years later, Mike Gracious from Stanford University showed that these regions within the default mode network also form a network. That is, these fluctuations are correlated at rest. And so this is particularly interesting network because it's not just motor, visual, auditory, things where we really know a lot about these systems already. This is a much more cognitive network and one that really hadn't been studied very much at all. And so I think that drew a lot of interest into looking at these resting state networks. In addition, Mark Rakel, who's a very prominent scientist, member of the National Academy of Sciences, was going around at that time and really promoting the idea of looking at the resting state, that there's a lot more there that is yet unstudied. So in summary, in addition to task-related neuronal activity, we have spontaneous neuronal activity that occurs actually even on top of the task-related neuronal activity. Both of these cause the hemodynamic changes that we can detect with MRI. So in the next lecture, I'll be looking at different pieces of evidence why these resting state functional connectivities do indeed have a neuronal origin.